Chong, and all the organizers of this wonderful meeting. I've really enjoyed my trip here, and the science is outstanding, and thanks for all those who supported this great meeting. What I'm going to talk to you today about is some of our work studying cardiac progenitor cells as a potential tool for therapy. First, I'll share some of our work and ongoing studies using induced cardiac progenitor cells, reprogramming to make those cells. And secondly, I'll share some of our recent unpublished work looking at ways to prime or stimulate cardiac progenitor cells for enhanced maturation using a compound called polyinosinic polycididic acid. So, first of all, why would we think about using cardiac progenitor cells and reprogramming them? And the justifications and the interest in doing this are similar to why people thought about induced pluripotent stem cells, except if you're cardiocentric like we are and you want to cut to the chase and get to cardiac lineages, it may be beneficial to have induced cardiac progenitor cells, or ICPCs, to do your studies of cardiac developmental biology, to look at cell biology and physiology, to model cardiac diseases as we've seen with iPS cells, for drug discovery or toxicology, and importantly for this meeting, for applications in cardiac regenerative medicine, because potentially induced cardiac progenitor cells would be less tumorigenic than pluripotent stem cell derivatives. They can be scalable, multipotent for the cell lineages present in the heart. Ideally, they can proliferate in vivo, so the grafts can expand. And in the perfect world, they may be able to migrate to areas of injury and enhance repair. So this work was now published a couple of years ago originally. Pratik Lalit was the graduate student in my laboratory that largely led these studies. And just to schematically go over the strategy, Pratik took a NKX 2.5 EYFP reporter mouse made by Dan Gary's lab a number of years ago that expresses EYFP in the early developing cardiac progenitors, and then that turns off as the first cardiomyocytes are formed. So if you get EYFP positive cells, it's a potential indication of a cardiac progenitor. And he crossed that with a mouse that allowed the transgenes that we express to be turned on by doxycycline. And he created a library of various cardiac relevant transcription factors, infected fibroblasts from a variety of sources, cardiac fibroblasts, lung fibroblasts, dermal fibroblasts, and then he added doxycycline and looked for YFP positive colonies, isolated those colonies as potentially reprogrammed cells, and then to see if they really were cardiac progenitor cells, Pratik carried out a number of assays in vitro. He looked at their differentiation to form different cell types and saw evidence for cardiomyocytes, smooth muscle cells, endothelial cells. If he transplanted the cells into a developing mouse embryo at about E7, at the stage E7.5 of the cardiac crescent, as the embryo developed, he could see the cells incorporated into the developing heart and developed into cardiomyocytes. And also he tested whether they could repair the injured heart in a mouse infarct model. And so he published that in 2016. Deepak Srivastava's lab came out with a similar cell population, although reprogrammed in a different way by transiently inducing pluripotency factors initially. But these induced cardiac progenitor cells were developed by ultimately using 5 factors in our hands after starting with a much longer list, and that's MESP1, TBX5, GATA4, NKX 2.5, and BAF60C. And when we did that in the presence of culture conditions optimized to allow these progenitors to proliferate, and that is by turning on Wnt signaling and inhibiting JAK-STAT signaling, we could get YFP positive proliferating cells that expressed a variety of factors you might expect in cardiac progenitor cells, including NKX 2.5, GATA4, and interestingly, they were mostly present positive for IRX4 as well. And so when Pratik took these cells and formed aggregates like embryoid bodies and then differentiated them in serum-containing medium, plated them down, he could see At the histology level, evidence for cardiac actin, alpha-actinin, and a variety of myofilament proteins suggesting cardiomyocytes could differentiate. In a smaller set of cells, he could see smooth muscle myosin heavy chain suggesting smooth muscle cells and some CD31 positive endothelial-like cells as well. And if he took these cells and delivered them in a mouse permanent ligation model of the left coronary, and in the low power views here, you see a nuclear staining and dense infarct because there's almost no cardiac actin. And then we zoom in on these areas of the boxes. You can see some of the GFP positive cells that label his induced cardiac progenitors and their progenies, co-label for cardiac actin, suggesting myocytes can form from those transplanted cells. Likewise, he saw evidence for smooth muscle differentiation of the cells and some endothelial cells that differentiated from the cells. And ultimately, delivering these after myocardial infarction in this mouse model resulted in dramatically better survival compared to the placebo, which was perhaps a little too aggressive since about all our mice died after 4 weeks, making it hard to do some of the functional comparisons. But at least at the mortality level in this initial study, it looked promising. So in the sort of setup for this study, we suggested that maybe these cells would be less tumorigenic, but we hadn't really tested it in our original study. And so this is an assay where we did a simple teratoma assay in immunocompromised mice, transplanting mouse ES cells compared to ICPCs. The ES cells put in the left hind limb flank here show a large tumor formation where the ICPCs didn't if we delivered 1 million cells there. In none of 12 animals treated. We could, if we looked histologically, see some progeny for the ICPCs transplanted there. We saw some smooth muscle cells and some endothelial cells. We couldn't find any cardiomyocytes, but probably not a great place for cardiomyocytes to differentiate with the signaling milieu there. But at least we didn't see evidence for tumorigenesis, so potentially a benefit of this cell source. Also, Ruben Alexanian, who is another student in the lab, wanted to see if these cells could be beneficial in generating new cardiac tissue using decellularized mouse hearts, taking a technology first published by Harold Ott and Doris Taylor, where they decellularized whole hearts by perfusing with detergents. And then rinsing that out. So a scaffold is left of extracellular matrix, and then delivering the ICPCs labeled with GFP. We could see the cells survived for 4 weeks with GFP fluorescence seen throughout these recellularized constructs. And then if you did histology on these and immunofluorescence, you could see that many of the GFP positive cells stained for cardiac actin, suggesting cardiomyocytes, smooth muscle actin, as well as CD31, suggesting we could get at least trilineage differentiation of these cells when delivered to the decellularized mouse heart. And to show evidence for function, we did optical mapping. D. Lang did these studies in Alexei Glukov's lab, where he, in this case, loaded the cells with a fluorescent calcium indicator dye and then did field stimulation. And you can see there's not even calcium transients throughout this decellularized constructs, but there are clearly areas that show calcium transients in response to the pacing. And so evidence that you have action potentials and areas of recellularization with functional cardiomyocytes. And if he did his optical mapping in a little different way, and instead of doing field stimulation, he did point stimulation and stimulated the recellularized heart with an electrode here, he could see propagation for several millimeters of the calcium transient, suggesting that there are coupled integrated grafts that form in these recellularized hearts. So we're encouraged that the ICPCs in a number of different models show potency to generate functional cardiac tissue. So I've quickly told you about our work with induced cardiac progenitor cells, that we can reprogram fibroblasts with 5 cardiac factors and get induced cardiac progenitor cells, that these cells can differentiate in vitro to cardiomyocytes, smooth muscle cells, and endothelial cells. If we put them in mouse embryos, I didn't show you that data, but they can also integrate and differentiate. If we deliver them after MI in the mouse heart, they improve survival and show potency to form the major cardiac lineages. We don't see evidence for tumorigenesis in these initial assays, and they can recellularize, decellularize heart tissues. So that's story number 1. Story number 2 is something that we're excited to present, and this is a study motivated by Mitch's realization that cardiomyocytes derived from human pluripotent stem cells are developmentally immature. We've heard about this a little earlier from Michael Laflamme, and that may be limiting some of the clinical applications and also the basic research applications as investigators want mature cardiac tissue to study drug effects, for example. And this immaturity is marked by a number of things. The electrophysiology of the cells is altered. There's more automaticity. There's a smaller IK1, the inward rectifier current. The upstroke of the action potential is slower. And there's also decreased contractility compared to adult myocytes. And metabolically, as Jay has mentioned, there's differences in metabolism, and these iPS cardiomyocytes initially are more glycolytic than oxidative in their behavior. So Mitch, in part, Mitch Bierman is a talented MD Ph.D. student in the lab who took on this challenge, and he learned from the Progenitor Cell Biology Consortium, the earlier version of this group, when he was at a meeting and heard John Cook present his work looking at activation of the innate immune system to more efficiently reprogram to iPS cells. And in that study, he learned about polyinosinic polycididylic acid, which is a double-stranded RNA mimetic that is thought to mimic activation of the innate immune system in cells, largely by binding to toll-like receptor 3. And so Mitch saw that this improved the reprogramming of iPS cells from fibroblasts and thought it may impact differentiation and the formation of cardiomyocytes and changing the timeline of that. For reasons that still remain a bit mysterious to me why Mitch came up with this hypothesis, but he developed a protocol where he first stimulated Wnt, and this is based on the GWE protocol, that's a monolayer protocol to differentiate cardiomyocytes, and then inhibited Wnt signaling, and at day 3 to 5 of that protocol is when the cardiac progenitors are formed, and Mitch tried to add this polyinosinic polycididylic acid, or poly-IC, at day 3 to 5, and then see what happens to the resulting progenitors and cardiomyocytes. And so the short story is that the poly-IC priming did impact at the progenitor stage, just adding that compound, that simple FDA-approved compound, for 2 days, when you looked at 30 days of differentiation, resulted in larger cardiomyocytes, whether you look in a 2D culture, so the cells are significantly larger, feature potentially of maturation. And if you do lane patterning on soft substrate, which Brett Napowicki and Wendy Krohn's lab did, you could also see that the cells became larger on average, have an increased length to width ratio consistent with more mature cardiomyocytes. So a couple of ways to look at cardiomyocyte size were impacted by just that 3-day treatment with poly-IC. And if you do electron microscopy imaging, you can see that the sarcomeres are more organized after poly-IC treatment at day 30. You see longer sarcomeres and more I bands in the sarcomeres that you observe. You can also look for myosin light chain 2V, a marker not only of ventricular lineage cells, which we believe the majority of our cells are in our protocol, but also a marker of maturation, as the first faded ventricular cardiomyocytes don't really express myosin light chain 2V. And we can see at day 30 we get many more, about 73% of the cells are double positive for cardiac troponin T and myosin light chain 2V, suggesting that there's an acceleration of the maturation process. although by day 80, they both plateau out at about 90% of the cells are MLC2V positive. So some acceleration of maturation, we believe, by this molecule. If we look at another myofilament marker, and this is cardiac troponin T, this is a little harder to look at. We collaborated with Inga and Veggie Kai, who did the proteomics with us to look at both adult and fetal isoforms of cardiac troponin T using top-down proteomics. And basically what they found when they used this technology is that the ratio of adult to fetal isoforms increases at day 30, but because the isoform switches in cardiac troponin T happen at a later developmental stage than the isoform switches for MLC2V, you see that this difference continues to expand for up to 120 days in culture with the poly-IC treated cells showing evidence for more adult-like cardiac troponin T or a more mature phenotype. If you look at contractility with optical flow analysis, which we did in collaboration with colleagues at the University of Kansas, Dana Asay and Andres Zorek, we saw that there is an increase in the mean divergence per frame without a change in the rate of contractility, suggesting that the contractile behavior of the cells increases as well. And if we looked at optical action potentials, the upstroke or rise time was quicker, and there was more oxidative metabolism of the poly-IC treated cells. So how can this happen? Mitch did RNA-seq analysis, and he found, interestingly, that the gene that was most greatly upregulated following the 3-day treatment of the cardiac progenitors was the Notch ligand, JAG1. He did an array for a variety of Notch genes. He found that the majority of Notch signaling-related genes were upregulated, shown here. And if he then used a gamma secretase inhibitor to inhibit Notch signaling, he saw that the no poly-IC compared to poly-IC treatment increase in cell area was blunted by DAP signaling, consistent with Notch signaling being integral in this biology. And finally, how can poly-IC activate Notch signaling? And I don't have time to go into all the details, but Mitch believes that it's an epigenetic effect, in part because poly-IC is known to downregulate type 2 HDACs based on previous studies. So Mitch did chromatin immunoprecipitation studies, CHIP qPCR, looking, for example, at H3K9 acetylation at a variety of cardiac myofilament promoters, as well as the JAG1 promoter transcription start, and he saw that there is an increase in this activating mark at all these sites, whereas other epigenetic marks such as H3K4 trimethylation isn't changed, nor is H3K27 trimethylation. So we believe that there are epigenetic effects impacting the cells early after poly-IC treatment that change the developmental clock present intrinsically in these cells and accelerate the maturation process. So does it matter for our potential in vitro application or in vivo applications? And so to look at this, Mitch took these cells after or without poly-IC treatment and delivered them to the kidney capsule of an immunocompromised mouse, and then basically looked a month later for graft formation. And what he found is that he got much larger grafts of more mature appearing cardiomyocytes after poly-IC treatment or with poly-IC treatment than without, much smaller grafts, and the cells are much more elongated, organized after the poly-IC treatment relative to not being treated. So we think this has potential benefit for accelerating the ability both in vitro and in vivo for the cells to differentiate and mature. So to summarize, poly-IC accelerates the differentiation of cardiac progenitor cells and the subsequent maturation of the cardiomyocytes, and we see that by a variety of parameters that I described. Poly-IC's effect is dependent on Notch signaling, and we believe it's one of the first interventions shown to modulate the cardiac developmental clock. And finally, poly-IC priming of cardiac progenitor cells results in larger human pluripotent stem cell derived cardiac grafts, and we think this may have clinical relevance for increasing the efficacy of this type of therapy. And I'll finish by acknowledging I've been fortunate to work with many great investigators at the University of Wisconsin, including those that I've acknowledged, as well as many great collaborators elsewhere, including Jay, the co-organizer for the meeting, and Ninad Bursek, who's also here. So thank you very much for your attention.
I think we've got time for one quick question. Yep.
Tim, I was interested to look at those decellularized hearts, and you know, the original promise of that type of experiment was to see if there was developmental information embedded in the matrix. Do you see any self-organisation into vascular structures or any other macromolecular tissue architecture when you seed your decellularized hearts with progenitors?
Thanks for that question. What we see most clearly is organization of the cardiomyocytes. So the cardiomyocytes that form like to couple to one another. We see traces of what may be capillary-like networks. We don't, and we see some endothelial-like cells lining some of the former blood vessels that you can tell by the lumens, but we don't see, at this stage, generation of a whole vascular functional network. So there's plenty of work to do.
Thank you.